All right, welcome everybody today to the Learn With Lowell show. I'm your host, Lowell. As many of you know, we talk to experts, scientists, and artists every week. Today we're joined with George Church, which is interesting in particular because this marks when this episode is going to go up in the beginning of March. It will mark the beginning of my 5th year doing the podcast, and George was there during the first year. So I think that's pretty cool. And George has a huge rap sheet that would probably take me an hour and a half just to say it. And people should know it, but I'll have like a screen rolling down his history and background. But George, welcome back to the show.
It's great to be back. Thank you all.
Yeah. So first question is, I was sitting wondering, what is it like to be you with all the things that you're doing and all the pressures that you have to do a great job, even just the stuff you put on yourself? And so I was wondering, how often do you think, and just like take a step back and think about how you went from like just a regular scientist to the point where you are now, where the measure of your impact is in millions, if not billions of lives over the course of like 10 years, 20 years, like the impact of your work is just so significant.
And maybe that's one of the purposes of humility is so you don't think about that stuff too much. I certainly do feel a responsibility to take, whatever education and opportunities have been provided and turn them into something that's, pay it forward.
Mm-hmm. Yeah. And then I think that's a great opportunity to jump in with a fan question. So I'll just read this verbatim because I suck at paraphrasing. So George is involved in multiomics, gene editing, gene therapies, epigenetics, AI, delivery. Like I said, huge rap sheet. Do you think that we have the whole picture or a relative whole picture of biology, or do you think there's still unknowns to be discovered in terms of biology, rejuvenation, that type of stuff?
Well, I'm guessing the question is about big unknowns. So there are obviously millions of little unknowns to be known that will get people excited. I mean, millions of things that will get the public excited even. So they're not that little, but really big things. I would have to say that a lot of the really big things in the future will be synthetic rather than analytic. That is to say, there's a finite number of things to be discovered, and we discover them kind of in order of their importance. Like importance could be abundance of a species on the planet, or pathological impact on humans, that sort of thing. And so what happens, it becomes kind of a successful approximation where we've gotten everything. But in terms of synthesis, there's no limit. I mean, until we know how to, use synthetic biology to create new universes, we're not anywhere near done. So I think there's a lot of big surprises in the future, and some of them will be reading, and some will be writing. A lot of them will be writing.
There's a great Neil deGrasse Tyson quote where it says, like, the more you know, the more the surface area of your ignorance expands. And so is there an area in particular that you feel, not in a negative way, but like ignorant of, like you're like, wow, like there's so much I don't know about it in terms of like, biology and synthetic biology?
Yeah, I mean, my ignorance surface area is expanding at the speed of light, I think. I think the biggest thing we don't know anything about is, and that includes me, is extraterrestrial life. It has the biggest delta between there may be zero to there may be hundreds of billions of life planets with life like ours and not like ours. So I think that's probably the biggest unknown. I don't think we'd be completely shocked to find that it's zero, or completely shocked to find out that it's trillions of stars, planets. But the details will probably surprise us.
Yeah, I feel like the weirdest thing is if we were to go out into space and then and it's not like so far where you, I think like mathematically, like if you go out far enough, you start seeing the same things as ourselves or something, like how the math works. But if we go like the next star system and like everyone looks like the same as us, that'd be so weird. It's like we must have been like seeded somewhere or something.
Yeah, well, panspermia is a likely mechanism. We know that there've been, many thousands of rocks that have passed between Earth and Mars over time. Some of those may have contained early life forms, some of which may have survived. But But yeah, if we go far enough away, the chances of contact are extraordinarily small, and so they'd be independent.
Mhm. And then, so you have many different projects that we, have alluded to. Is there something that you're working on right now that you're particularly excited about? Like you have so much on your plate, but at the same time, like not even like, yeah. Is there something in particular that you're working on that you're like, wow, this is really cool? Like you're like really like the difference between like I'm like checking boxes versus like you're really excited about developing something.
Yeah, I have a tendency to stay away from checking boxes and
Yeah.
and incremental stuff if I possibly
Mhm.
can. Yeah, every now and then a previous revolution, either from my lab or some other one, just demands applications or incremental, big incremental
Mhm.
things. And in fact, some of our biggest our biggest changes have been, the product of lots of increments. For example, a reduction in cost of sequencing by 20
Mhm.
million fold was the product of a bunch of two folds. Maybe came in every few weeks or months. But to your question about what it's a popular question, is like, which of your kids do you like the best?
Yeah.
I would say probably the one that is most impactful is, when the genie gives you one wish, you ask for more wishes. And so the
Mm.
equivalent for science is asking for more years of research,
Mm.
more youthful, healthy years. And so we have a number of related projects, both in my lab and in my alumni companies, that are aimed at longer, healthier years. So that's one. Another one that I've been excited about for decades is recoding multiplex editing and recoding of genomes so that we can, among other things, make organisms that are resistant to all viruses. And that finally paid off this year. We have the first example of an organism that we think is resistant to all viruses, and we tested this by doing field work, getting a bunch of new, thousands of new phages, and having very sensitive assays for replication, and found none. So that's another one, and there are plenty more. Getting a code for differentiation, in other words, a code that tells us how to get to any place in development, both young and old, backwards and forwards. I think we're making progress on that, so that's very exciting. Sequencing
I don't.
everybody on the planet is another one, and I'll stop there.
Yeah. How with the improvements in sequencing, what would be the cost of sequencing 8 billion people? Like, is that actually like doable?
It's totally doable, and the cost. Let's say the cost of an item depends on how much you get back, right? So it's like you get a kickback or something like that, or you have an investment and then you get a return on that investment. And my estimate right now is the genomes are about $300 each now for a very high quality diploid genome, which is not the original genome project, which is not diploid. Anyway, it's about $300. It'll probably be $100 within a year or two. But the return on that investment is on the order of $10,000 or more on average. So for some people, you'll get nothing back. For other people, you get $1 million back in savings for the healthcare system, whether that's a government system or insurance system or some combination. That's the return on investment. So actually, it wouldn't cost anything to sequence everybody on the planet. It would result in a net gain. And I think there's a growing amount of economic modeling that supports that.
Yeah, it sounds like the Apollo program, where people always say like, "Oh, we put a lot of money into it," but the return on investment was like, for every $1 we got like $12 or $13 back.
Yeah.
Like that's a great deal. Just like you don't notice it in the moment because people are like, "What aboutism about everything?"
Yeah, I mean, so that even though we haven't gone back to the moon, and you could say, well, that was nothing, that was a wash, it was one of the most expensive projects in history, right up there with the Manhattan Project. But we did, even though we didn't go back to the moon, we got GPS satellites, we got weather satellites, and a variety of the telecommunications satellites. So those three alone were some of the biggest ticket items in return on investment. So
yeah, and then the making organisms so that they can't be like virus proof is like how I'm gonna summarize it. What was the organism? I don't think I read about this. Was it? Did you get in pigs?
It's a bioarchive preprint that will be a Nature paper soon. Been accepted. The organism is E. coli, which has a particularly severe virus problem. There are a huge number of characterized and uncharacterized viruses. In contrast to like the second favorite industrial microbe, I would say, is yeast, baker's yeast. And baker's yeast has essentially no virus problem. I mean, that's just evolutionarily. So E. coli has enormous one, yeast has very little. They're both the top among the top two industrial microbes. But I would say that most industrial microbes fall into the E. coli category. I mean, they have virus problems. So, for example, almost everything that's in the dairy industry, yogurt, cheese, so forth, all those microorganisms have virus problems. In fact, that's one of the reasons that one of the first applications of CRISPR was in the dairy industry, because it had such a big virus problem. Still not solved, but it was a motivation. Yeah.
I'm still waiting for them to put up the Pasteur statue, the giant milk thing that we talked about like 5 years ago on because you're on, I think, like the road that's near. I hope this isn't doxing like where you work, but like there's like a road nearby called Pasteur Street or something, like the pasteurization guy. And I think like 5 years ago we were talking about like we should get a giant like milk carton to like symbolize like his street and everything.
My address at Harvard Medical School is Avenue Louis Pasteur, and we do have a giant milk carton, not on Avenue Louis Pasteur, but down in downtown Boston. It's in some tourist section, where they have a few museums, the Children's Museum and the Computer Museum. I'm not quite sure what the milk is all about. I think it's a place where you can get ice cream.
They should move it down to your street, given what the Vis is up to.
And a giant wine bottle as well while they're at it. Yeah.
And for so with viruses, how do we know that the viruses just like won't see that as a potential food source and adapt to find it? Like how can we be certain that they won't just make new viruses?
Right. So the way we did it is we for 2 codons out of the 64 triplets, we swapped the amino acid from serine to leucine. So normally these two would be serine, and now they're leucine. And the host, the E. coli host, was in on the game, and so we swapped them around so that the host is completely unaffected. The virus was excluded from this. Normally there would be coevolution, where for every step that the bacteria would take, the virus would take this, a complementary step and would keep up with it. But we took it offline and made thousands of changes, about 20,000 changes. And now the virus has, every time it sees those 2 codons, which is quite frequently, these 2 serine codons are common in every protein. That means every protein has a mixture of serine and leucine at every place this occurs. So that means every protein is broken in multiple ways. And so the only way to get them back to where it's at all functional would be to have some kind of way of editing all those sites, this kind of the same way the host was, without intelligence, and not edit all the other ones so it's dead. And the level of mutagenesis that you would need to get that level of editing would kill it in other places. So it's hard for it to escape. Now, there is the possibility of making new viruses. So viruses, in a certain sense, are as little as a polymerase that it got from the host, encapsulated in some way in proteins plus lipids, or plus or minus lipids, And so that could happen. But I think it would take a long time. I don't think we've ever observed one going from scratch. If one did arise, then we would, maybe there are things we can do to prevent the polymerases from being co-opted, and we also don't know how capsids form spontaneously, but probably there's all kinds of ways to aggregate proteins, and they just become more and more symmetric as time goes by.
Yeah. And then I think, like using the software analogy, like potentially if there was something like that, you could just offer like a software update to the biology, like anyone who's using that type of thing. And then it would, like, you could always stay out of step if there was a virus. It was like,
absolutely. Yeah.
Oh, were you say something?
Nope.
Okay, good. I just always like to make sure I'm listening. When you develop technology like this, I feel like there's got to be like 100 companies that just kind of like follow you around and just like wait for it to come out, and then they buy up the IP. To what extent is like, not that like bother you, but in the sense that like, if everyone's, I think in like the 70s or 80s, like there's like this myth that like there was an electric car and like some oil people bought it up so that no one could have electric cars. So how do you make sure that like no one just buys up the IP and like puts it on a shelf? Granted, like I guess if you sell it to like Ginkgo Bioworks, like the E. coli stuff would just fit into their program and then they would just love it so much. But how do you make sure that like the IP actually goes to good people, like that are actually going to use it?
Yeah, that's an excellent question. There are two ways. One is that Harvard usually almost always puts a clause in the agreement that says if you don't meet the following milestones, the IP returns to Harvard, and they don't return your money. So it's Harvard's advantage to monitor it and make sure that they do meet the milestones, and the milestones are usually crafted to be pretty easy to observe. The second way that one avoids this is by spinning off companies rather than being reactive to companies on the outside buying it. You spin out a company that is formed by the postdocs that invented it, and they're highly motivated to see it succeed because it's their entire life up to that point. Their entire professional life has been developing this technology, and if anything, they're biased in favor of it, where they think it's the greatest thing, where it may or may not be. But a big company, when it buys up stuff, it's because a lot of big companies have lost their ability to innovate. They'll even admit this. I've been inside such big companies as a board of directors, and the way they innovate is by buying companies that have already innovated. So basically, they don't see it coming. When Harvard says, "Hey, we have this IP available," they don't see the value of it the way that a postdoc that invented it would. And even if they did, Harvard's going to tend to favor the young startup because eventually it will get out either on its own or by being acquired by one of these big companies at a later date.
Makes sense. And then when people are transitioning from academia to the startup, like the postdocs, like narrowing in, since you're in so many different projects, are there problems? Are there things that they need to adjust? Because like the academia from I haven't worked in academia, but I'm told it's kind of like it's one mode of success, like how to do something. But then startups, like it's entirely different. They have to be faster, like whatever. What transitions or what new skills that they have to develop to make that jump successfully, so they can build something and without it like crashing and burning?
Yeah, I think that's a great question, and I think some environments are better for that transition than others. So over the years, we've accumulated a bunch of alumni that have done this successfully, and they either inspire from a distance or up close. They will inspire the next generation and tell them where the landmines are and where the pots of gold are, and that all helps. It helps them know whether they want to go into join a big company, form their own company, or stay in academia or something else. And then, so if they have the prepared mind, that's a big part of it. The main thing is that academics are the job is to spend money, and companies' job is to make money, and that's a big cultural difference. Also, there tends to be a lot more deadlines and milestones in companies, which isn't necessarily a bad thing. More productivity, a little more focus on incremental improvements rather than radical risk taking. But each of these is, it's some people like it one way, some people like it other. Some are willing to go through the academic in order to get to the commercial stage. Companies don't typically train graduate students, and so you have to kind of pass through this academic phase at some point or another. It's like metamorphosis, but I think it works out overall. Now, one thing to be particularly careful about is when the entire company is made up of academics. In other words, there's often they're fine. A scientist postdoc will find a business person, either mature or just finished business school, and then they'll make a symbiosis and then make a company. That's healthy. But sometimes they'll want to have their buddies be CEO, CSO, CTO, et cetera. And that also works, but it's much more challenging. It's like elephant balancing on a ball. It definitely can be done. In fact, I would say most of my companies are of that second type at this point, but it took a few years to get to the point where we could do that routinely.
I have a friend who, when he left postdoc, and people may know him, I don't know, but when he left, he tried putting like doctor people, like postdocs, on sales calls, and it was like, he was like, why did I do this? I'll just hire salespeople, because like how a scientist thinks about conveying risk is different than like how you convey risk if you're like really trying to understand what people want. Right. And so that like he lost a lot of money. It's like it's a different skill set. Though I do think the structure of questioning things is transferable to sales. I think the people just need to like think about it a little bit more. I think maybe they'd figure it out.
It's good to have communication throughout the organization, especially when you're small. People will wear multiple hats at the startup phase. They'll be both human resources and CTO, for example, or something like that. But it's tricky. Yeah, it's tricky.
Sometimes it feels like this is a great time to be alive because there's so much development going on. And we're talking about longevity, and for the most part, a lot of the longevity interventions are geared for like as early as possible, or it's like cholesterol. So it's like, you can do that at any point, but assuming probably as soon as you have a cholesterol problem. I'm thinking of like O'Connor and what he's working on. But do you feel like your age grouping tends to get ignored in terms of longevity, in the sense that like most things aren't, like I think most people are like shooting for like staying in like the 30s-ish range, like getting everyone to like having like the mind and body of a 30-year-old. So then it takes time to like group it out. So do you feel like in your age grouping, and I think I have a vague idea how old you are. I think you're like, I don't want to say in case I'm wrong, but
68.
You don't look as old as
68.
Yeah. Like the thing is like you don't look the age, So it's like you're still doing great. Like if someone said you were like 45, I'd be like, yeah, okay, I get it. But do you?
My father went white when he was 19, so I was a little bit later, so maybe I'm a little younger.
Yeah.
Anyway.
Well, it's also a benefit having the white hair. I think webe people like. We're like, hey, this guy has a point. Then they don't realize how old you are, like, because sometimes people are like, oh, you're young, you gotta wait your time. But so for developing longevity technology, do you see like kind of a shift? From my point of view, it seems like people are more geared towards like the 20s to like 40s, but I don't. And then there's like some for like cholesterol, etc., from like 40s to 60s on up. But like if I were to like make a bell curve on it, I would think like the largest amount is like in the younger grouping. So then do you feel like for your age grouping that you're kind of being like left out? And then are you developing technology to like account for your age grouping?
I didn't think about it that much from that point of view. Partly because I see a lot of drugs being developed for late onset Alzheimer's, for example, which is 70, 80 years old, which is older than me. I see a lot of heart disease and cancer is still mainly affects people at end of life almost by definition. And the end of life is moving up there. We've basically been adding 1 year of life to every 4 years that human race is around. And we've now doubled the average lifespan over the last 170 years. So, but we need to do faster than that if we're going to save people alive today. It has to be 1 year per year, not 1 year per 4 years. And I think that's happening because it's like the exponential is really starting to take off in biotech. And I think it's hitting, first it hit reading and writing DNA, which is very molecular, and which is not that far away from like electronics. But now it's hitting into cell biology, developmental biology, and they've got their own exponential, which is similar. And I think that will impact. And finally, the biggest thing that makes me not worry about what age group is being favored. This is kind of like, I am worried about ethnic ancestry groups and gender discrimination in research. But with aging, you got this big thing, which is aging reversal. And we've seen plenty of examples of it in animal systems, in human cell culture. And it's a real thing. And so that changes the discussion as to what age group you're looking at. In fact, my ex-postdoc and co-founder of Rejuvenate Bio, Noah Davidson's group, just published a paper where they applied the therapy, a gene therapy, a triple gene therapy. Almost all our gene therapies for aging are multiples. Applied that at 124 weeks of mouse age, which, to put that in perspective, about half of the cohort had died from old age by the time the other half got the treatment, and it showed a very significant increase in lifespan. So it's within our power to aim for very late in life treatments. There's kind of two schools of thought: is that you need to treat early in order to extend life. The other is that maybe pretty late you can reverse things, and so I'm keeping my mind open on those two schools.
Yeah, there was a great series. I think it's the Calico series, where for the longest time they just would, like, every 20 years they'd get in like this. They'd go into like this pod or something, and it would rejuvenate their body back to like 20, and then they would age up to 40, and then they go back to doing that again. And then sometimes I wonder to what extent is that something we're going to be doing. Like, we don't cure Alzheimer's, but we can run rejuvenation up to the point where you basically don't have it functionally. Kind of like how some nuns are so active up until the point they die that we don't realize that they had like plaques and stuff going on in their brain. I wonder to some extent, is that what we're going to do? And then eventually we can cure it, But I wonder about these things. What is your position on that? Do you think we'll have like rejuvenation and then eventually cures for the illnesses, or do you think we'll just jump rejuvenation in the sense that I'm talking about it and have cures for the different illnesses? Which is
again, there's a few schools of thought here that are not mutually exclusive. One is that there's serious damage to DNA, RNA, and proteins, and you have to go in there with your pliers and fix them. The other is that if you just convince the cell epigenetically, you change the culture of the cell, it will think it's young, and then it'll get its own pliers out and fix itself. Now, there are obviously some things that are hard to fix by oneself. For example, if you delete both copies of a tumor suppressor gene, you've lost that information from that cell. In principle, you could get it from another cell, but that mechanism hasn't been shown to be common. So those you might have to literally go back and then reinsert the gene, maybe put in a few extra copies. And that could be done preventatively or reactively. I think that I fall on the camp where I think that most of it is epigenetic, and if you change the epigenetics either naturally or unnaturally, you can fix a lot of things. And we're getting better and better at delivery, so we can deliver extra copies of tumor suppressors preemptively. So, I think it's going to be a mixture of things that deal with specific symptoms of aging and things that aim at the core mechanism of aging. And hence, once you get the core mechanism, you can reverse it. So I think we're going to see both in big time in the next few years.
Has there been anything in the last 5 years, just thinking of it from the bookends of first interview and now, that has surprised you that has come out? I think the first time we spoke, CRISPR in terms of therapy was but they're still developing it. And I think this year they came out with the first 2 CRISPR therapies. I believe like that people are actually going to be using them. So I think that's been a change, but I think that's like an expected change. Has anything like surprised you in the last 5 years? Either longevity and any of the other areas we're talking about?
Definitely not surprising. In fact,
yeah,
everything about CRISPR was not particularly surprising, other than people's reaction to it. That was surprising to me, because we had really great editing prior to CRISPR, for which Mario Capecchi and Oliver Smithies got the Nobel Prize for 1980s work. It just wasn't that efficient. So, normally, I don't think making things a little bit more efficient is such a big deal. If you make it 20 million times more efficient, like sequencing, that's a big deal. But CRISPR was maybe 4 times more efficient or something like that. What has been really surprising? Well, I was really pleasantly surprised by how easy it was to edit repetitive elements. So, with all the best editing methods, it was very hard to get above, say, 70% editing. So, 70% of the cells would have 1 edit, right? And so if you think, well, to get 2 edits, you're going to have the square, and 3 edits, it's the cube. And pretty soon, none of your cells have all the edits that you want, right? And so the idea of making dozens of edits seemed unlikely but nevertheless, we had to do it for the pigs organ transplants. We had to get rid of the viruses that they produce. All pig organs produce retroviruses, and retroviruses, we were a little the world was a little gun shy about retroviruses, because that's what screwed up gene therapy around the year 2000, is that the retroviral therapy caused cancer via the LMO2 oncogene. And so the FDA did not want to have a bunch of pig retroviruses infecting human cells in immune compromised patients. Anyway, we did that, and our first experiment, we got 62 at once, 62 edits, each of which individually seemed like it was, 50 to 80% probability. So it just seemed astronomically unlikely that, it's like that you would, pull up the ace of spades 62 times in a row. And then since then, we've extended it. We've now done 25,000 edits. And that I'd never cease to be amazed at how easy it is actually to make that number of edits. And I think we're going to see more and more uses of multiplex editing, just like we have more and more uses of multiplex therapies, or polypharmacy is sometimes called. So for engineering agricultural species, industrial species, and even human cells for cell therapies.
Mm-hmm. I think I was watching an interview with you. We were talking about how like you're working, or like there's theoretically we could get to like 1 million cell edits at the same time with multiplexing, and that's I guess like the timeline from like 20 to 20,000 is it seems pretty fast. So then 20,000 to 1 million doesn't seem like it'd be that far off.
I think we may already have the protocol. We just haven't applied it yet. Mm-hmm. We have definite applications for it, and we're moving in that direction. I mean, for example, the virus resistance that we have in E. coli only took 20,000 edits. The virus resistance in mammalian, the equivalent virus resistance in mammalian cells would probably be hundreds of thousands of edits, maybe 1 million. So that's one driver. De-extinction of genes, not necessarily species, or maybe even species, could involve millions of edits. I think we're going to get away with hundreds, but since this is moving so quickly, I wouldn't be totally surprised if we get to millions by the time we need it.
Is there I don't know, I've been playing Kerbal Space Program. It's a game on like space flight, so everything's in like space terms for me, which is bad. But an apogee is like the top point before you start going down. Is there an apogee where like if you did more, like where you have like reduced returns for how many more edits you can do. So like if you had like, 10 million, the ability to affect 10 million cells at the same time, versus like 1 million is like getting the extra difference in 10 million doesn't actually do much more. Like you reach like a cap where like you're not doing that much more, if that makes sense. So like, is there a cap where like editing more cells at once would actually not be useful, or is it more generally as useful?
I just want to point out there's two kinds of multiplexing here that you nicely pointed out that I had glossed over. There's one is the number of edits per cell, and that's the thing that I was talking about. We're at 25,000, we're heading towards 1 million. And then there's a number of cells edited at once, and you could have both, which is a large number of cells edited, a large number of positions each. So as early as 2009, we made billions of cells that each had a small collection of edits. So it wasn't one edit per cell; it might be 5 or 10 per cell, but billions of cells. So we had a very large diversity of genomes that were designed, not random, not entirely random. I think that points of diminishing returns, certainly for the, let's say, codon remapping, there's a certain number of edits you have to make before you can delete the transfer RNA or swap the serine and leucine that I mentioned to get you virus resistance. And if you stop before that, you're going to have a key protein that's got a mixture of serines and leucines, just like the virus, and so the cell's going to die. So that's a threshold thing. For de-extinction, let's say, there may be as few as 100 that allow the elephant to be cold resistant and to do its thing on restoring the Arctic, the vibrant Arctic ecosystem as a keystone species, maybe as few as 100. But it might take millions to make it genetically identical to its common ancestor or something like that. And that would be a point of diminishing returns where the ecosystem impact, you got most of it in the first 100, and the only reason you keep going is because you can. Right.
Makes sense. Yeah, and then I definitely see an application for I'm going to call it the make it so like viruses can't impact animals, like testing out on agriculture so that you don't have those problems wherever, because like for the most part, viruses come from animals, like when they hop over to humans, and then that's what kills humans because it's not built for our body.
Yeah.
Which anyone familiar with the show will know that I comment on like how when the Europeans came to America, we brought horrible stuff here, but there was not really a transmission from Americas back to Europe because the Americans didn't really have that many domesticated animals. I think they only had alpacas that they really domesticated. So there wasn't that many, like Europe had like pigs, chickens, they had so many different things. So there was just many more opportunities for it. So making agriculture something where that jump doesn't happen, then we don't potentially won't have like, COVID and stuff that happens to us. You talked in one of your recent interviews about software as biology. And you made a comment, and I was like, oh, I wonder what he means by this, because like you weren't able to expound on it. So I thought I'd just ask you now. You stated that using that as an analogy, you say that there is a lot of biology in analog circuits that people are overlooking. And whenever you say, hey, someone's overlooking something, it's like, oh wow, I definitely want to learn more. What do you mean by that?
Well, I mean, there's two opportunities. One is making cells more digital, and the other is using what is the strength of cellular computing, which is more analog.
Okay.
And examples of analog is, measuring the temperature, and you can turn that into analog to digital, and digital to analog are very common in electronics. And the same thing happens in biology. Is you can say you build up a certain amount, and then you decide where it is in the day, and you set your circadian clock. Or you get a certain temperature and you decide that it's spring and it's time to do the whole vernal cycle thing, et cetera. So, but then there are other things where there isn't a switch that's flipped. You stay analog, where the temperature increases a little bit, then you stop shivering, and you get rid of your thermogenesis brown fat stuff, and vice versa. So there's no, I don't know, hard and fast rule where something has to
Mhm.
be digital, has to be analog, but there definitely are cases where it's a little more efficient from an engineering standpoint or an evolutionary standpoint. One thing I'm keen on getting the digital going back in is recording information into the DNA of cells. We've set a record of recording 2 terabytes of information into the DNA of mice. About, you can record physiological and developmental data, and we think we can scale that up to 20 petabytes by hiding the information in the parts of repetitive elements that we will find empirically are not disruptive. So the reason I describe it in petabytes rather than analog terms is because DNA is essentially digital, and it's got 2 bits per base pair. But a huge fraction of biology, the thing about biology, it does this very natural gradation between analog and digital. So, for example, a group in UC Irvine has used our method to encode hypoxia data from analog state into this digital DNA state. So, and we need to be able to go the other direction too. So we need to be able to take the digital DNA data and turn it into analog physiological responses.
Yeah, I think what uses? Well, one I can think of is like, sometimes we want to put like biobanks or something on the moon, or in different spots. But if we could just like record all the data and like hide it in cells, or like a really like power, like like the people make comments like if there's a nuclear war, like cockroaches would rule the world, so we could like hide that stuff in like cockroaches for us to pull out in the future. But that's like more like apocalypse type stuff. What uses is there to have like 20 petabytes of data stored in cells?
Right. Okay. So, I mean, there's a fork here in the road where you're storing data that is definitely digital. Let's say, tables of where all the cities are and things like that. That's digital information. It's very cultural. The other is where you're storing data that is biological, that was essentially biological analog and digital data. You're storing it in a new form that's more compact. And so you're essentially creating a time record, kind of a tape recording or a flight recorder is, I think, the best analogy, where in case something goes wrong, you can go and inspect the record in whatever cell you want. So if something went wrong in the liver, you can go into the liver and itself and see what its flight recorder says. So I think this could be used medically or potentially environmentally to if something bad happens, you go in and you can go to a particular coordinate where the bad thing happened and lift out some DNA and figure out what happened. You don't have to sequence the entire ecosystem or the entire organism to figure it out. So it's just like a flight recorder, I think, is the main use case that I have so far. But it's one of these things, a little baby, you don't know where it's going to go next. So,
yeah, that would be interesting if it was like if you had it in every organism on the planet. So whenever you were exposed to it, you could kind of like you could see like what was happening to it for the course of its life. It's kind of like tagging sharks. You could just take a little biopsy and then you'd have all the information. Then it'd be really cool.
Exactly.
The one thing that you in one of your other talks today I wanted to touch on before I think we jumped in more longevity related things is you talked about using synthetic biology as manufacturing. Well, we talked about this in our first one. I don't know if anything has developed any faster on this, but you talked about it as a way to alleviate poverty. If we came in and we could build, manufacture through synthetic biology medicines, then that's one less thing that the society that is in poverty has to worry about, so they can invest in other things. But I was reading that in like Poland, for instance, they use clams to determine if water treatment plants have filtered enough of the water for it to be drinkable. They use like a biology process. So I was really taken by this idea of using biomanufacturing to alleviate poverty. If you could go in and then build water treatment, like self replicating and rejuvenating system for like water sanitation and all these other things. Because when poverty, one of the biggest things is like they don't really have clean water. Medicine's a huge one, of course. So if you could alleviate all those things, and then make it something that they just have to like, feed, I guess. That may be really cool. I was just like really taken by that idea of, can we use synthetic biology to alleviate poverty and lift people up and build systems? Because like even in America, we have like Flint, Michigan, that has like, I think it still has lead in the waters, and we have like chemical spills and all these other things. So like having like a biological process that could break it down, and then or a manufacturer to create something, because biology does seem extremely efficient compared to like what humans can do. Like I don't think we're as efficient as what biology can do yet.
Yeah, so you're touching a nerve here in a good way, in that I think that not only can we create a virtuous circle for poverty, where you alleviate a little bit, and that gives them a little bit more health and education to make the next increment in lifting up out of poverty, lifting themselves out of poverty. In particular, I think that one can imagine, and we're working on a box essentially where it produces food, and you do complete recycling. So you can think of the house. You can have a house that's like a space station or a submarine, completely sealed in, nothing exchanged with the outside world other than energy from geothermal or solar or wind, whatever. That then helps recycling, and because you're in complete control, each household, it has its own system. Once you have a clean system that doesn't have lead in it, doesn't have other industrial pollutants in it, it will stay clean. And also, you will have known mechanisms for cleaning it up in the first place. So there are known mechanisms by which you can biologically decompose dioxin and other toxins, where you can take heavy metals and sequester them into a lump in the side, and you just leave it there. But then from then on, it's internal. You eliminate supply chain costs and insecurity. So a lot of the problem with poverty is not so much they're impoverished today; it's that the next time there's a supply chain issue, they have to pick up and go, and they become refugees and all kinds of things like that. So you need to have a more stable supply chain, and what could be better than having it, all within your house. Also, a huge amount of the environmental damage is done due to making pipelines and roads and manufacturing of trucks and so forth, and that could be eliminated if 90% of the manufacturing occurs in your kitchen.
And even some of the basic stuff that people do for like biohacking, or just like when they're first learning genetic engineering, is like, hey, can we add something and then have like it like be bioluminescent at the same time? So if there's like something going wrong in the system, like it could be as simple as like color coding it, so you're reducing the level of technical expertise that a person using the system would even need to have. It's kind of like the Chinese box. Like they would just need to know like these colors correspond with these things, that inputs they have to put into it. They just go on with their day. It's
even better than that, in that you can make it so they just ask what they want. So they say, I want, a vegan burger, right? And you press vegan burger, and out comes one. And so there's no colors to match or anything. Like you just see what you like, or it even remembers what you like, to have on Fridays. And it's kind of like GPS, on your phone. Most people have no idea of how many satellites there are and what angle they have to be at, and how they're synced up with 37 atomic clocks, and all this stuff that is required for it to tell you to turn left. And that's the beauty of it, is you don't even need to have simple programming skills. You just you talk to it the way you would talk to a extremely knowledgeable person. So I think that same thing will be true for manufacturing. It's not that hard to recycle, complete recycling. We have plant and animal systems that have been going for 8 decades in a completely sealed container. It doesn't have to be completely sealed with humans, but it would be, if you can do that, then you have a whole new set of capabilities. Even submarines and the International Space Station, rarely go more than a few months. They're always getting resupplied, so it's not really a sealed system, except for those short periods of times that are in between supply runs. But we can definitely do it. We know how to do it for animals.
Mhm.
And it would also reduce certain existential risks. So, certain kinds of, depending on your sources of energy, certain kinds of, asteroids, pandemics, super volcanoes and so forth. You could, and you could also prepare for leaving the planet if you test out the whole survival in small clans or small isolated systems. You test it out on Earth where the risk is lower. If something goes wrong on Mars, you got a couple of years to get back to the nearest emergency room. While on Earth, you test out these modules right in the shadow of a city.
Mhm. I was watching this, it was a Smarter Every Day. He's a great YouTuber. He like goes and discovers things. He went on a submarine and he looked at like how they produce food, and they had like this, like a corridor stocked with food. But to get to something, they have to take it all out to get in there to find like, ketchup or, maple syrup. And they've organized as best as they can, but there's not that much space in a submarine. So instead of needing like, so many different food stocks, you could just have like several food stocks and then use this biomanufacturing process to then create the food from there. Exactly. So I think that like even like it'd be space efficient, and you don't have to do like play Tetris with it too much, which would be pretty cool.
Yeah. And in fact, it's doing the Tetris because it's probably all the different producing industrial microbes are inside the box, and you just say make this, and then somebody else has programmed it. It adds a whole new level to the idea of recipes, right? Where the input is something much closer to raw synthetic biology.
Yeah, I think the closest to this type of system that I can think of is like cellular agriculture in terms of taking cells and just producing it based on what you're giving it. Is there anything that you can think of that's working towards this type of system?
Well, that's the disappointing thing is there aren't. I mean, there are some algae that are fairly nutritious and tasty, like spirulina. That also, but no one has. There's almost no recipes for making things entirely out of such organisms. Usually, they use them as garnish or a little bit of seasoning just to feel good about it, but. Having recipes entirely based, but it's not that far away either. I mean, making recipes is something that everybody can do. It's one of the few citizen sciences that literally everybody participates in one way or another. So I think this would just be incredibly enabling to be able to. It could keep track of the nutrition for you. You just tell it what you like to taste, and you experiment with new. Sources of metabolism.
Could so the interesting thing with like energy generation, for instance, is it's all basically just built on a turbine. Like even like nuclear power plants. I thought that'd be cooler. Like people were like, oh, they're gonna like kill people or whatever. And I actually looked into how how they work. It's no different than just like spinning a turbine. Like just like steam is generated, whether it's like coal or whatever, and a turbine is turned, and then like that's power that comes out. Could we ever like? Obviously, you shouldn't do this because it doesn't make sense. But like, if we like took mitochondria and like blew it up, and I guess that's a horrible analogy, but can we use a biomanufactured energy plant to produce energy as efficiently? I think nuclear is the most efficient form of energy. So can we do the same thing with power using biomanufactured like a cell thingamabob? That's a technical term.
Probably, yeah. Well, take photovoltaic as an easy example. I'll get back to nuclear. Photons come in, they get electronically converted to a voltage, and then that can go straight into biological systems. So you can have either electro metabolism, or it can turn back into photons that are at the right wavelength. So photons coming in include infrared, green, and ultraviolet, none of which are well tuned to a lot of photosynthetic systems. Anyway, so you can essentially remap without turbines Then the biology has all kinds of chemistry. Basically, almost all chemistry probably could be done that we're interested in could be done biologically. Nuclear would be, I think, more challenging, but there are thermonuclear devices in certain space probes that this is past tense, this is not in the future, that do not involve turbines, as far as I know, and so they're not at the very high level of a modern fission reactor. And we don't even know what the fusion reactors are going to be like, but probably they're so hot that it's going to be very tempting to run turbines. But yeah, I mean, I'm not. I don't think it really matters if you have a centralized facility. Turbines are fine. It's just when you want to be off the grid, then you might want geothermal or solar. Probably something geothermal might be the safest one, maybe wind, because if you get like nuclear winter or something like that due to a supervolcano or a meteor hitting, then you want to have something that's a little bit safer than photovoltaics from that kind of standpoint.
Recently been reading about
geothermal is effective almost anywhere on the planet. It's easiest to do when it's near the surface, like in Iceland, but everywhere on the planet has, 600 degrees centigrade if you drill deep enough. It takes some investment to get down there, but then from that point off, you're off the grid.
Yeah, I was recently reading how efficient geothermal is in terms of how easy it is to hook up, because you can actually get them in for houses and stuff, and they'll last for 50 to 100 years, like the piping that you put underground, and it's not even that deep. I was thinking you have to really be in a special spot. Maybe it's from sci-fi because it's always in a volcano we're doing geothermal or something. But a lot of the systems we're talking about, it reminds me of this science fiction. But there's subgenres like frostpunk or but basically there's biopunk where everything's built out of biological systems, and it feels like there could be a future in the not too distant future where a lot of the things in society are built using biology versus people's hands, in terms of us having to build it, which would be pretty interesting. And if you do want to do stuff like that, you should build it in the Midwest because it's much cheaper. I tell everyone this all the time. If you look at how expensive things are on the coast to the Midwest, it's so much more affordable out here. Though it's really cool that this can be built where it is, and they make use of all the other networks around it in terms of innovation and stuff like that. There was a fan question about the woolly mammoth, and I just want to pull it up real quick because it's a really cool thing. I think when we first were talking about it, it was just not like a twinkle in your eye, like you were working on it. I think you had there was a book where the guy, I think it's called Woolly or Mammoth, where there was he talked about how there was a mouse and it roared like a lion, not a lion, like the woolly mammoth, because you put some cells into it, which I think is just really great imagery, even if it wasn't true. In regards to the mammoth restoration project, can you talk about the modifications that you're going to be making to the Asian elephant, other than giving it fur? I looked online and I tried to see if I could reverse engineer what was actually being done. But is it possible to talk about what type of work you're doing on the Asian elephants to move it to the woolly mammoth in terms of in specificity? Or is it
sure,
secret?
absolutely. It's we're going to have a paper or two coming out pretty soon where we lay this out. Maybe within the next 8 months or so. And we've discussed some of this, but the list is mainly focused on cold adaptation, or you could say handling both winter and summer, where the winter is -40 and the summer is normal. And Asian elephants actually can tolerate pretty low temperature and enjoy playing in the snow and even playing in frozen lakes. They'll break through the ice and they'll swim around for a while, but they can't do it for very long or for very cold or very low, high wind chill. And so one needs fat deposits. So there was 10 centimeters of fat pretty much all over the body of the mammoths that was not in modern elephants. Instead of the short, spiky sparse hairs that are essentially radiative. They radiate heat. Instead, the mammoths had the thick woolly hair. So those two probably aren't that hard. There are more physiological, more biochemical things, like their hemoglobin was adapted to release oxygen at lower temperatures than is normal. So the skin temperature is getting close to freezing point. Even though the core body temperature is the same as our core body, the skin is different. So that's one of the genes that has been restored, de-extincted, and shown to be have plausible functionality. The ears have to be smaller so they don't get frostbite. A few things like that. There's some things that may not be present in the mammoth that's also on our list. They're present in neither of the modern elephants nor the ancient ones, and those are things like resistance to killer pathogens. In fact, back to the whole virus thing that we talked about earlier, elephants have a very serious virus problem. They're endangered species because of herpes viruses, and so we have various strategies we're exploring to make them resistant to either all viruses or to this specific EEHV virus, which is part of what's making them go extinct. And then we may have adaptations to, poaching, where they either have very short or no tusks under certain circumstances, and in other circumstances where they're well cared for and guarded, they'll have the big, maybe even bigger tusks than usual. So those two things, the virus resistance and the tusk manipulation, may not depend on the mammoths, but most of them are inspired. The other place where we could deviate a little bit from the mammoth inspiration, we could look at polar bears and penguins and other cold-tolerant animals for ideas, and maybe make these elephants even more cold-tolerant than the mammoths were. But the point is, we're not limited, and that's the short list that you were looking for, I think.
Yeah, is it for controlling the tusk length? Would it just be like if you gave them like a certain food size? I think of bees, like the difference between a queen bee and a regular bee is like they're just fed something differently. Would that be like how you'd regulate it in terms of like being in a safe spot versus not a safe spot?
Exactly. We already have projects where we've made organisms that are biocontained, meaning that if they don't get this chemical, which is not available in the wild, it's only made by organic chemists, then they can't replicate. And in the case of the elephants, it might be that same chemical, which they can't make tusks without that chemical. And so you put that in the feed in a very well protected animal reserve. It might be, hundreds of square kilometers, but it's very well guarded. But then when you let them out into the millions of square kilometers of the Canadian and Alaskan and Russian Arctic, their tusks will be small, and that's already been shown. There are tribes of elephants that don't have tusks, and they do fine. There are tribes of elephants that have ridiculously long tusks, and the genetics of this has been studied, and we'll exploit it.
Is there any plans to expedite the gestation period? Because I think elephants have like 22 months, which is a pretty long time.
Yeah. We are definitely interested in short gestation periods. It'll probably result in smaller animals, although there are animals that keep growing, in other words, they're born small, but they keep growing until they're quite large. For example, the record for short gestation is in marsupials and in rodents, non-marsupial eutherian mammals. It's around 20 days for a mouse and maybe 13 for the fastest marsupial. But the marsupials are essentially born, they look like fetuses. They can crawl up and get to the milk, but they And the same thing's true for the rodents. When the rodents are born, they're blind, they're hairless, they're very, very fragile. They're not like cattle. They come out running.
Yeah.
So there's a trade-off, but it would be helpful to get feedback quickly by having a short gestation. And I don't think we're going to make a 20-day gestation period rather than 22 months, but we might. I mean, there might be some reason to do that. Yeah, and but more important maybe than gestation length is how many we can do in parallel. And I think if we can do this ex vivo, which we've got a fair amount of effort on that, then we can have them, essentially in this giant warehouse or conveyor belt or something where where we could have 10,000 of them growing without interfering with the reproduction of the current endangered species.
We're close to being able to replicate gestation outside of the womb. I thought we only had like artificially could like test drugs on like it's like a little chip. I didn't know we were close to having something where you could like really take it out of the system, out of the mom.
Well, it's hard to say how close they are, but there are parts that make it seem like we might be close if you're embedded in the field. So for example, you can take mice up to almost 9-10 days, which is halfway through gestation from fertilization, and human preemies we can take them so they're only halfway through a normal human gestation and take them all the way to birth, and they're fine. So in a way you can get halfway from both sides. So it seems like all you have to do is bridge it. The problem is that most of the protocols for getting halfway from fertilization, you haven't really hooked it up to umbilical cords and placenta properly. So you can't just trivially swap into preemie mode. And there's just a little gap there. So that's one way of looking at it. Another, we are getting just much better at producing organs for transplant. I mentioned the pig organs as one of them, and keeping transplantable organs alive outside the body. So if you can keep the endometrium, the uterine wall, alive like you would an organ outside of the body, then implantation is almost automatic. And then the two halves, the embryo half and the maternal half, have well-evolved mechanisms for all the feedback, physiology, and so forth. So the key thing is getting the endometrium to be able to be healthy outside the body. The development is fairly much on autopilot, but no one. It's amazing how little effort has gone into maintaining organs outside the body, mainly because most transplants don't need to be outside the body more than a few hours. So there's not a heavy, up until now, there hasn't been a heavy motivation for setting records on that, but that's what we're doing.
Yeah, with organ transplant, I've always felt like the time horizon, like they have to move really fast. But there's also a lot that just like there's no one around to use them, so no one gets to use them. So having like a biobank of organs that can just be maintained until they're used. But then again, then just like manufacture our own organs is also pretty cool. So I'm generally curious if when you're developing a product, sometimes you just like find people and you see what their pain points are, and then people can like kind of help in building things around that. I'm curious for you and your innovation, are there problems that you have that if someone's like, hey, I come in with, I guess this like the ex vivo process to generate elephants would probably be a process you would love help with. But are there tools or techniques that you wish were being that you wish you had to do the work that you're doing to like expedite them?
Oh, absolutely. Most of the tools I wish I had, I'm working on, but still, some of them seem to take a while. I would wish we had better microscopy. Right now we finally have microscopy that can work at pretty high resolutions, like 20 nanometers, maybe better, where we can label DNA, RNA, and proteins. So that's a way of, when we're engineering, you want to be able to check to see what you're doing, right? And so a lot of what we're doing is invisible with the naked eye, and so we need better and better microscopes. And the problem is that once you get to super resolution, if you want to do both large scale and small scale at the same time, you end up with a lot of data. So petabytes just to do ordinary things. And then another thing that doesn't scale well, and so a lot of this is scaling, is say 3D printing. There's a lot of things we would like to be able to 3D print anything. I actually teach a course called How to Grow Almost Anything, which is it's on the internet, it's international, and it's a sibling course of how to make almost anything. And we would like to be able to merge those so that we can make any device that is currently inorganic with biology, and then use the two together to, we should be able to make that. And biology is fairly good at inorganic materials as well, but it's not really there yet. So I'd like to have and to use developmental biology rather than Cartesian 3D coordinate printing, which is very slow and doesn't scale well. Biology has trillions of printhead equivalents per cubic millimeter in the form of ribosomes and cell division and so forth, all these molecular machines. So it has this incredibly high density of simultaneously performing printheads, while most 3D printers are, kind of this simple, Cartesian robot. So that's a partial list, but the list goes on of things that are missing, but they're getting filled exponentially. So I'm not panicked, but it doesn't fill itself by itself yet. So it requires a great deal of ingenuity and hard work to fill those gaps in technology, which is kind of the main thing my group does.
Yeah. It's always great to read what you're up to. I wonder if you had things we're not covering and we don't know anyone who's covering, like if you had like a list and then people go, oh, okay, they could like use that to generate some ideas. But I'll link the courses in the show notes. So fan question. Gene therapies that have been approved for rare diseases are very expensive. What should we expect in terms of cost? I don't think you'd be able to answer this, but cost for gene therapies targeting. Well, it's for Rejuvenate Bio, which I think is, what should we expect in terms of cost for gene therapies targeting the biology of aging, like those developed by Rejuvenate Bio? Because I think like you can guess even at this point. This is pretty early. I think
this is a great question. Is this a fan question? Did you say?
Yes.
Okay, it's a great question. And I'm ready for it. Okay, so I have pointed out that there may be a different strategy for rare diseases and common diseases, and I would put aging in a common disease category. Another common disease is pandemics or very large vaccination programs. Even if they're currently rare diseases, we still have to vaccinate the entire population. And what happens is, for these common diseases, infectious and age-related, is since everybody is a consumer, the denominator for, you have kind of fixed costs for R&D and clinical trials, and the denominator is the number of people that benefit. So for rare diseases, that denominator is tiny. It could be 100 people a year. And so the cost is, the highest cost I've seen so far for a therapy is $3.5 million per dose for one of the recent rare disease gene therapies. And in fact, that caused me some concern when I entered the field of gene therapy, many years ago. Is I've been the proponent of making technology equitably distributed, so that even poor nations and communities would have access. And I realized, and we did that. We brought down the cost of reading and writing DNA by 20 million fold and improved quality at the same time. But for pharmaceuticals, they seem to be creeping up rather than going down exponentially. But then if you reanalyze, sort of to think about it a different way, the recent COVID vaccines, the top 5 of them were all formulated as gene therapies. And one category of those 5 is adenoviral capsid around double-stranded DNA encoding the vaccine protein. And that got down to as little as $2.18 per dose. So down from 3.5 million for a rare disease to 2. And they're both, viral capsids around double-stranded DNA. They're gene therapies. So I think that's where it could go. I think that's going to apply to aging reversal and aging reversal, or age. If you don't like aging reversal, then aging disease, core treatments for hitting multiple aging-related diseases, which is what we're doing. That could be in that range. Now, I'm not making a promise on the behalf of my companies they're going to sell things for $2 a dose, at least initially. But it's certainly, we've got the proof of principle that works. It's been tested. These gene therapy-like things have been tested in billions of people worldwide, and I think the experience has been fairly positive. So I think we're off and running now on common gene therapy. So now, what about the rare ones? I think one solution that is getting better and better every day, and was already pretty good, is genetic counseling. And this can be done either pre-conception or premaritally, which is the best time, because there's no risk of false positives causing damage on their own. And it can be done as late as in vitro fertilization, or even prenatal. The in vitro fertilization is getting much better. One of my companies, Orchid, is sequencing every single embryo that goes through the IVF clinic. And that helps not only avoid these very serious but rare diseases, you get them all at once because you get the whole genome, but it also helps with the fertility itself, because a lot of failures in the IVF clinic. So on average, you have to do 5 rounds of hormone treatment and IVF to get one child. And a lot of those are due to aneuploids or other genetic diseases that are so fatal they kill the child before it can even be born. And so if you're anti-abortion, you should be anti-spontaneous abortion as well. And this is a way, by sequencing the IVF embryos, you can see which ones are going to survive. And we might get that down to 5, from 5 IVFs per baby to 1 IVF per baby. So I think all of that is going to handle the rare ones. Now, IVF isn't quite as equitably distributed as the genome sequencing, which will be basically free. And once you get the return on investment worked out, and aging reversal drugs, which might be once in a lifetime and $2 a dose. So, and that also could be free, because it would be to the government's advantage to have people, working past retirement age, being healthy, not consuming. So I think those are the different answers for rare and common, but they both hopefully will end up with equitable distribution to even the poorer communities.
Mm-hmm. Something that I thought of when you're saying that reminded me of the making it so the viruses can't affect things. Because you were talking about you mentioned COVID and the mRNA vaccines.
I didn't mention mRNA vaccines. I actually mentioned adenoviral capsid double-strand DNA. But mRNA vaccines are also formulated as gene therapy. It's just a less common one and a new one.
I was wondering, just as a quick aside, is it possible to modify a virus to have a gene drive, so then when it goes out in the wild, it just infects all the other ones, and then they die really quick? Could you do that? I don't know if you could. I was just wondering.
Well, so just for the listeners, gene drive is typically something that spreads through sexual species and ideally ones that breed quickly. So these would typically be insects or rodents. It doesn't work as such in viruses, nor in asexual bacteria, nor in asexual plants and animals. Viruses already spread pretty quickly. I guess the question is, can you make a virus that will outcompete the other viruses, or when on the rare case that viruses exchange genetic material, will it win in some sense? I think that's challenging but possible that you could. But the problem is that getting viruses that outcompete natural viruses sounds a little risky, and I think rightly so. It's not going to be easy to fund, and it should certainly, if it is funded, it should be done in glaring sunlight with a lot of people watching, and a lot of different kinds of voices being heard about whether it should be done. I think much more powerful than a gene drive in a virus, or the equivalent in a non-spectral species, is the vaccines, is preparing our immune system for these viruses.
And I know we're coming to the end. There's a
also that we could do recoding, which is the ultimate, because that makes us resistant to all viruses rather than one at a time via vaccines, some of which our immune system just doesn't remember it very well. Our immune system doesn't remember coronavirus or norovirus very well. So the multivirus resistance we get from recoding would be much more awesome, also much harder to get implemented.
Is that something, if we were to do like 10 years out, is that something you, like if like, so woolly mammoths 5 years ago was something that you were developing with mice, and now you're developing it in the actual animal itself. 10 years from now, do you see stuff like that being something that would actually be in development in terms of like for humans? I'm curious, like, what do you see for the next 10 years, I guess?
It depends on your definition of development. It is in development already. So I can with confidence say it'll be in development 10 years from now. But, I think that we could, if the rate at which we can do multiplex editing, 25,000 today, maybe 250,000 tomorrow, a couple of years from now, we could construct cell lines that are multivirus they're resistant to all viruses, just like we did for the industrial microorganism. And then we could use those cell lines to make cell therapies like hematopoietic stem cells, neurons in the brain and so forth, which are already happening in a non-multivirus resistant way. So you could say any cell therapy or organ therapy or transplant would be better if it were multivirus resistant. So you just pop that in as an option. It's like, do you want bucket seats? Okay, check. Okay. And 10 years from now, I don't know where it'll probably still be in development. It is true that some clinical trials are now happening in 1 year rather than 10 years, but until that's very common, I hesitate to say anything takes less than 10 years in medicine.
So then one of the listeners wrote in, and it's a very personal question, so I appreciate their courage in asking it. They said, I have a disability called CRPS, and mine is a result of an amputation and reattachment, resulting in multiple pain issues due to nerve damage, inflammation, other issues going on for the last 12 years. Is CRISPR something that could potentially fix issues like that, or is that something more like that would CRISPR be good for, like regrowing an arm? Basically, we're wondering, like, what could CRISPR and genetic engineering technology do to alleviate the type of suffering he's going through? Or is it more like, of the two branches, would it be easier to just regrow a limb, I guess, would be my way of summarizing it.
Yeah. Complex regional pain syndrome is a subset of pain in general. And I think there's been a breakthrough in pain in that we have pathways now that are non-opioid pathways, non-cocaine pathways, that are based on sodium channels that actually reflect a person or a set of people in the world that have chronic insensitivity to pain, or CIPA. And so those could be harnessed. And those people are not doped up the way you would expect of an opioid or a cocaine derivative. They're fine. So and you can make it local or you can make it temporary. And I think that's happening. It could be small molecule drugs or it could be gene therapy. I don't put CRISPR on a pedestal, even though I did have something to do with it, making it out there in the world. I think it's just one of a set of tools. And if you're going to be making transplantable limbs or organs, then CRISPR is not really the centerpiece of that. I mean, most of it is, biological and tissue engineering, and you're going to use a variety of editing tools to get you there. If it comes from an animal, then most of the challenge is making it compatible immunologically and physiologically. And I think we've handled that right now. We'll know soon. We've got 2-year survival in preclinical non-human primate trials. So that bodes well for getting it into human clinical trials very soon. I think the insensitivity to pain was probably something that's better done with drugs than with transplants. But there are plenty of cases where we will need to transplant limbs, as well as, unfortunately, there's still a lot of warfare in the world. There's, occupational and car accidents and so forth. But yeah, my guess is the pain will come from progress in the sodium channel pain pathways. And then completely separately will be organ and limb transplants.
And then are there books that you've read over the last couple of years that you'd suggest people check out? I know your book, basically all the longevity books, but the one thing that I'm doing is I'm compiling all the books that everyone's recommended in 170 plus episodes, and I'm putting in a PDF for everyone to read. So this will be like kind of a nice bookend. But are there any books you recommend people check out? I think the first time we spoke, you were a big fan of what's his name, Hitchhiker's Guide to the Galaxy. I think we quoted that back and forth a few times.
Right.
Yes, yes. But is there anything else? Anything you recommend people read, either on aging, on biotechnology, just anything that you've enjoyed?
Yeah, I have a tendency to read books that are written by friends, and they ask me to write a blurb for the cover of the book. So I have kind of a bias. And then things that are complete fiction, totally off topic, which is my wife and I read in an Audible stream when we're doing chores together. I'm writing a book. Hopefully that will be
cool.
on your bookshelf for your 200th episode. But yeah, I don't have anything in particular that I think there's a need for more books on the revolution that's occurring in synthetic biology, manufacturing, aging reversal, and so forth. Probably that will be more than adequately filled in the next couple of years. But right now, I think most of the books on synthetic biology are, like biofuels, not terribly useful, and the ones on aging are, how to eat slightly better things, which again is not exactly news. I might mention a few new brand names, but they're really not revolutionary. I hope I'm not insulting any authors or anything, but I think we're just at this interesting transition zone where if you want to write a book that's not totally science fiction, you have to stick to things that are a little bit boring. In a couple of years, science fiction will become fact, and then suddenly all the books will become exciting. And it's not the fault of current or future authors. It's just the nature of scientific revolution.
I think that if you were to do like a crowdsource Kickstarter type thing to write a book, and you could just write it, as advanced as you want, I think you'd get more than enough funding to write whatever you'd want. Like you can have like a team of researchers too. It's not
a financial problem. It's
okay.
It's more a time management problem. I do have, as you pointed out earlier, I do have a few things that I'm doing that limit my writing time, but it's getting done. It's getting done more and more quickly.
Mhm. And so, just like, what is like one fiction book that you're reading, or Audible reading?
Let's see. Um, there was one on a sort of Darwinian. It was the something of all things, and it was about a woman, a fictional woman, contemporary of Darwin, who had her own very similar theory, the signature of all things, and it was based on this principle that there was a signature that plants and animals would kind of tell us what their function is. It could be for medicine or something like that by their shape. That was part of it. It was much more complicated than that. It was a little lovely story. It wasn't. I wouldn't call it a science exclusively, but it had that little thread going through it that I thought was nice. There was one by Andy Weir.
The Martian or the new one?
The Martian, but there was another one, Hail Mary.
Yeah. He also has Egg.
What?
Yeah, it has. Sorry, there's a short story. I think it's called Egg, which is also pretty good.
Yeah. Anyway, I mean, those two are very creative, and I think they're very kind of pro science. You sort of feel at the end of it like that's great. You know, they think out of the box. They do stuff that needs to be done, and so forth. So I thought those were good. You know.
Yeah.
There's also a story of the dog barking at night, something like that, which was written from the standpoint of a autistic child, older child, and I just thought it was very well done. In that, it was a very interesting mystery, kind of a small, relatable scale. Too many mysteries, involve 12 murders, and this was, no people died, and it just seemed like it was more relatable. But even if you've never met or never known you've met an autistic person. And not all these books are brand new, but some books are so permanent, that they don't need to be brand new. Sometimes you miss it the first time through.
Yeah, I'm reading some Dostoevsky books, and it's like, oh, this is nice. I prefer like the more like War and Peace type Russian authors.
Yeah,
because they don't really have paragraphs.
Dostoevsky, yeah.
Yes. Thank you. I didn't think of couldn't think of the name. Yeah, it's also like it's a little like Dostoevsky's like he didn't know what a indentation was. It's a little hard to know like where his thoughts begin and end. But it is fun. So I agree with your point that you can go back and pick things up on the second time. So someone did suggest that you might be qualified to apply for Calico Labs' new like CTO position, and I was like, you know what, you might have the skills, but but I think you have enough things going on. I know we're going over, so then I'll I'll think of some books that would be good for you, and I'll send them to you. But then, so this was the Learn With Lowell show. I suck at outros, so I do this together. I just want to thank you so much for coming back on the show. This is great, and thanks so much for taking so much of your time, given like everything you got going on in your day.
Well, this is very important to have a conversation with a broad set of voices that can feedback through you. So I think happy to do it.